Hi, everyone. Welcome. Thank you for joining us today. My name is Monica, and I'm the Outreach and Recruitment Specialist in the Marketing Department. While we wait for everyone to settle in, let me tell you about UCSC Silicon Valley Extension. We are the Professional Development Division of UC Santa Cruz, and we're located in Santa Clara, California. We offer courses and certificate programs for working professionals to update their skills. Our courses are designed by experienced instructors who are actually working in their field, and one of them is joining us today to talk to you about the principles of drug discovery and development. Dr. Edward Rosen is chair of the UCSC Extension Biotechnology and Clinical Trials Design and Management Certificate Programs. Dr. Rosen has an academic background in virology and has devoted his career of nearly 4 decades to the study of viral diseases in academia and in the biopharmaceutical industry. His areas of research include the genetics underlying the viral mechanisms of disease and drug discovery efforts to treat HIV/AIDS, herpes virus, and several respiratory viruses. Before I hand it over to him to begin, we ask that if you have any questions during the presentation, you can submit them in the Q&A box at the bottom of the screen, and we'll go over as many as we can after the presentation. And if we don't get to your question, don't worry, we'll follow up with you after the webinar. This presentation is supposed to go from about 2 to 3 o'clock. We'll try to keep it as close to 3 o'clock as possible. And if there are any questions that you think of after the webinar is complete, you can email us and I'll put that information in the chat shortly. And then feel free to also put any comments and thoughts in the chat. If you want to tell us a little bit about yourself, what industry you're coming from, we'd love to get to know you. Just go ahead and enter that information in the chat. Thank you so much for joining. And with that, Dr. Rosen, I'll hand it over to you.
Okay. Thanks very much, Monica. So hi, everybody. Welcome to this webinar on pharmaceuticals. And I think I'll jump right in. Let's see. I don't have control about what's moving here. Let's see. I can't seem to. There we go. Whoop. Okay. So I don't know if you've ever had this experience or not, but certainly I have, when I was much younger, when I'd go to the pharmacy to pick up a prescription drug or maybe purchase an over-the-counter drug. Well, what's happening here? Losing my slides. Okay, here we go. So one of the experiences I had, in those days was really kind of having some thoughts about that medication I was picking up at the pharmacy. Where did it come from? How is it available, to the pharmacist for, for selling to customers who have prescriptions or an over-the-counter drug? How does it come about? And of course, beyond that, the key question I had, because obviously I had a condition or some symptoms, the question was, will it work for me? And I think that's, a pretty common question that most people think about. And then beyond that, you might be thinking, is this medication I'm picking up safe? And likewise today, because we're in the midst of the global coronavirus pandemic and a lot of discussion about the vaccine, the kind of questions you have about a drug, where does it originate? Will it work for me? Is it safe? Are also applicable to vaccines. So I kind of thought that today we would touch on some of these questions in a general sense. So in regard to topics for discussion in the next hour or so, I'm going to give you sort of a high-level overview of drug and vaccine development. And then I'm going to transition into really giving you some examples of some of the advanced technologies that have come about in the last several years or perhaps a decade or so that really relate to drug and vaccine development. And I'll give you some key examples about innovations related to antiviral drug treatments for the SARS-CoV-2 virus, so the virus that actually causes COVID-19. I'll touch a little bit on the technology of the development of the messenger RNA vaccines for COVID-19, which, as you know, were the first vaccines approved globally for COVID-19. I'll touch a little bit on some innovative treatments in the last few years related to treating cancer. And then I want to finish off the discussion by talking to you because a lot of my students and a lot of attendees for our webinars are really interested in opportunities for employment in some area or another. And my focus today will be on the biosciences. So I'll talk to you a little bit about employment opportunities in the biosciences in a general sense. And then I want to give you kind of my perspective, having been a hiring manager in the biopharmaceutical for many years, regarding some advice for individuals seeking jobs in the biosciences. And then lastly, I'll be happy to address any of your questions. So I want to begin with this slide, which really provides an overview of drug discovery. And basically, the drug discovery and development process is divided into two main periods. So there's a preclinical period that lasts 3 to 4 years. And when I use the word preclinical, what I'm really referring to is these are laboratory studies. So we're not dealing with any studies involving any human volunteers that would be participating in clinical trials on those investigational drug entities. And then the other major part of the drug development process is the clinical period. And the clinical period is that period that we're really focused on testing investigational drugs for efficacy and safety in human volunteers who are participating in clinical trials. So just to give you a sense of what really is involved in, the drug discovery and development process, we're talking about an investment on the part of the biopharmaceutical companies on the order of, 750 million up and beyond $1 billion. So certainly when we're talking about drugs that are drugs derived from recombinant DNA technology, like monoclonal antibody drugs, which has become very common in the last decade or so to treat a number of diseases, and especially cancers. Those drugs, typically can be well beyond $1 billion in regard to development. So starting over here, one of the first things that a company has to think about when they initiate a new drug discovery program is what disease do they want to select. So one of the key questions that they'll be focusing on is, does this disease satisfy the criteria for having a significant is it a significant unmet medical need? In other words, there might be drugs available to treat this disease, but are they satisfactory? Are they providing the degree of efficacy, the effectiveness of the drug in impacting the disease in a positive way, or do they have some safety side effect kinds of related issues? So companies will pretty much think about those questions relating to satisfying an unmet medical need in regard to initiating a new drug discovery and development program for a particular disease area. So kind of the second question that will come to mind for these companies is, what do we understand about the pathogenesis of the disease for which we would like to bring a new drug to market for treatment of those individuals with that disease? And so we think of this concept of target identification. What do we know about the mechanism, the process involved in the disease coming about and being maintained? So basically, one of the things that pharmaceutical companies will rely on is a vast amount of academic and perhaps some pharmaceutical research that really casts light on the process that allows the disease to come about, because that can assist in identifying targets for developing drugs that would specifically treat those diseases. So I'll go over this a little bit more when I give you an example of some of the drugs that are in development to actually target the SARS-CoV-2 virus. So then the companies have to decide, once they know the disease, once they know what target they want to focus on for their drug development program, is to initiate then a drug discovery process. So there's a lot of different means for companies to actually identify and discover new drugs. Some of them are somewhat random, by chance. Others are, highly thoughtful, very following very careful procedures. and so forth. In many instances, sometimes it simply relates to, generating, chemically synthesizing a drug, or using recombinant DNA technology to bring that drug to the fore. So one of the things many companies will do is actually end up with more than one drug candidate during the drug discovery phase. So one of the key goals then is to really characterize those different molecules that they have, that target the specific processes that will work against the disease, you know, um, coming to the fore, carrying on, and so forth. So characterize and prioritize that. The goal is to utilize some small animal studies where we look for the ability of the drug to produce efficacy in a small animal model of the disease. We'll look at the pharmacokinetic properties of the drug. How does that drug to be administered to an individual? Is it given as a tablet, an oral drug, capsule, caplet, or is it given as an injectable? So intramuscular, intravenously, and so forth. So the science of pharmacokinetics really provides information that allows pharmaceutical companies to decide how they want to administer the drug. And not only that, what happens to the drug once the drug is administered. So you can learn a lot of information about those properties of the drug in small animals. And likewise, ultimately, we need to do very carefully regulated, carefully controlled toxicology studies that really assess the safety profile of the drug in small animals. Now, certainly, how an animal acts in a mouse or a rat or a guinea pig of that sort does not necessarily predict how a drug will actually act in humans, but it certainly can give us some important advice about the possibility of safety associated issues in humans, as well as the possibility of the drug actually providing efficacy in humans. So ultimately, the whole goal then of the non-clinical studies that are carried out early in the preclinical phase of drug development has the goal of identifying a single investigational drug, sometimes referred to as a drug candidate, that we can test in human clinical trials. And of course, we start those studies with the what's referred to as phase 1 type clinical trials. And these are mostly focused on safety studies, but safety in truth in combination with pharmacokinetics. So to initiate these phase 1 clinical trials in human volunteers, the pharmaceutical company has to submit something referred to as the IND application. That stands for Investigational New Drug Application. And that's basically an application that will compile all the non-clinical data that the pharmaceutical company has, safety data, efficacy data in animals if it exists, in combination with how the company proposes to test the drug in the phase 1 clinical studies. So assuming FDA gives the green light to that in the United States, and by the way, for those of you that are calling in outside the US, there are FDA counterpart agencies pretty much in all countries across the globe that follow pretty much the same process in allowing companies to initiate clinical trials in human volunteers. So phase 1 studies, as I mentioned, focus mostly on the safety of the investigational drug. Companies typically start with very low doses and escalate up. And they hopefully will find a dose of drug that will provide a blood concentration of that drug that will provide a pharmacologic effect or produce a desirable effect to benefit the patient who's being treated for a given disease or condition. Now, in phase 1 studies, generally, but not always, we rely on individuals that are healthy individuals. They don't have the target disease. So reason for that is, bottom line, if there are some safety issues with the drug, it's much less likely to be injurious to a patient who's healthy as compared to a patient who's already sick and has the disease. So assuming the drug is safe, looks good here, then we move into phase 2 clinical studies. And when I say these phases, each of these phases can include actually multiple clinical trials. In the phase 2, we're very highly exploratory in the sense we're trying to establish how much drug do we administer, what is the dose of the drug, what's the duration that we would be administering the drug and that kind of thing. So the idea then is in phase 2 to actually test the drug for ability to produce a desirable effect in individuals who have the target disease, while also at the same time being able to demonstrate that the drug is reasonably safe in those individuals. The goal of the phase 3 studies is to identify a dosing regimen that can be employed in the phase 3 studies. So phase 3 studies are usually much larger in terms of the number of patients participating in the phase 2 studies. Here, we'll still be looking at the ability of the drug to be effective, to produce efficacy, as well as the drug to be safe to those individuals being administered the drug. So the bottom line is we always want to look at the benefit of taking an investigational drug in a clinical trial outweighs the risk to the patient, the so-called side effects. We also want to, in phase 2, ensure that the overall results are reproducible. The hallmark of science is reproducibility. So it's not unusual for a company to be required by the FDA and other regulatory entities to actually do 2 phase 3 clinical trials that both show safety and efficacy of their investigational drug. So once these trials are finished, all of the clinical data compiled along with the non-clinical data, and some non-clinical studies will occur concomitantly with the clinical trials, are compiled into the marketing application. So in the United States, the marketing application is represented by an NDA. This stands for New Drug Application. It is actually focused on drugs that are chemically synthesized, or a BLA, that's a biological license application that is utilized by companies who are developing biologic kind of drugs. So in other words, drugs produced through recombinant DNA technology, like hormone replacements, like monoclonal antibody drugs. The FDA then will review the application, and if the science is good and the drug looks to produce a benefit to the patient, provides a scenario where the benefit to the patient outweighs the side effects or the risk associated with the drug, then FDA would typically approve that drug for marketing. And just so you're aware, any company who is fortunate enough to get an investigational drug, advance an investigational drug to phase 1 clinical studies, only 1 in 10 investigational drugs that are started in phase 1 studies will actually make it to the market via the FDA approval process. So basically, there's a 90% failure rate. And so if you think about that in the context of the costs involved, companies could have spent $1 billion or more to get a drug to the point of FDA review, but if they're not successful in getting FDA approval, then in essence, all of this money is, I won't say it's wasted, but it's certainly not going to provide them an opportunity to recoup their research and development costs based on selling this drug because they don't have FDA approval. So one of the reasons that sometimes we think of newly approved drug as being so costly is that pharmaceutical companies are pricing drugs not only to recoup their research and development costs for their drugs that are successful, but they're also having to pay those costs associated with drugs that failed, those 90% of the drugs that made it to phase 1 that did not make it onto FDA approval. So I want to turn your attention to the vaccine process. It's very similar to the drug development process in the clinical phase over here, but there are some difference in the non-clinical activities. So obviously, when we talk about vaccines, for the most part, we're talking about infectious agents. And today, since I'm going to give you some examples related to the SARS-CoV-2 virus, the causative agent of COVID-19, I'm going to talk to you about vaccines for viral diseases. So company needs to select a particular viral disease that they're interested in. They have to determine a vaccine approach. So if you've been following the news carefully in the last year or so, you know that there are a number of different types of vaccines that have been approved for COVID-19. But beyond that, there are also a number of vaccines that have at least not been approved in the United States or the EU as yet, that are quite different than the current messenger RNA vaccines or the Pfizer vaccine. So the idea then is to determine what kind of vaccine do we want to make. So are we talking about an activated viral vaccine or a live viral vaccine where the virus is attenuated, so not able to produce disease? mRNA vaccine. Sometimes we can have DNA-based vaccines. We haven't heard so much about those as yet. We can add subunit vaccines. There's actually one in the works for COVID-19 that may be approved by the end of the year, and chimeric vaccines. So I'm not going to go into the details on these, but I just want to let you know that there are a number of different types of vaccines that can be developed. So companies will have to decide which one they want to go for. So then the process is to, do the research to generate the vaccine candidate. So we want to ensure in animals and maybe non-human primates that the vaccine will elicit a host immune response. And of course, there we're primarily looking at, antibody responses or T cell responses. And then ultimately, we want to show beyond demonstrating an immune response in the host against the viral vaccine candidate, that vaccine can also demonstrate safety. So likewise, the effectiveness of vaccine as well as the safety of the vaccine, which is tantamount to the same criteria that exist for developing drugs, is also relevant here. So then we want to move into the next phase, and that is conduct a challenge study. So if we're dealing with a viral vaccine, we would typically immunize some small animal, mice or rats, or maybe non-human primates, and wait for them to develop an immune response. And then we would challenge them with the actual infectious agents, in this particular example, the virus. And should those animals then be protected from, developing disease or serious disease and maybe death, then we know that the vaccine is effective in our animal study. So bottom line is we'll do some work in formulation to, enhance the immunogenic properties of the virus, so that when we move the vaccine into human clinical studies, we'll be successful in having human volunteers actually, initiate both an antibody response as well as a T cell response. So bottom line, as I said, pretty much this side of the slide is essentially identical to what we would do in drug development. So what's kind of interesting, what's really, I should say, kind of it really is interesting, is historically, until just very recently, the time to develop a vaccine has been about 10 years. So, of course, it's been quite astonishing to many people, scientists included, that Pfizer and Moderna vaccines were really, received emergency use authorization by the FDA about 1 year after the start of the COVID pandemic. So, quite a fascinating development, and a lot of you'll read in a lot of the press and conspiracy theories and the like as well. Any vaccine that comes about in a year is not likely to be very safe. So the reality is although the vaccine itself, came to the market within about a year after the start of the pandemic, the technology of using mRNA to program cells to make a vaccine has really been intensively researched for more than a decade. So a lot of the basic research that led to the success that both Pfizer and Moderna had in getting their vaccines approved by the FDA relatively rapidly, in about a year after the start of the pandemic, relied on that decade of research, setting the platform up for the development of those vaccines to really come forth and be available to the public in about a year after the pandemic started. So I just want to kind of emphasize that if you have an opportunity to work in bioscience today, it's an incredibly exciting time to be in any of the bioscience field. Now, obviously, my career has been, about half my career has been associated with working in the pharmaceutical field, bringing drugs, to the fore to treat a number of human diseases. But, from really about the last decade up to coming decades, I can't think of a more exciting time to have a job in the bioscience fields. Now, some of the discussion today, as you've seen so far, is kind of focused on biotechnology and biopharmaceuticals as a specialty fields in the bioscience area. But there's other kinds of research being done in bioscience, contract research organizations, academic labs, government labs, private institutions, and so forth, which all fall under the category of bioscience research. And certainly a lot of what these companies do is to do research on disease processes, provide really key information to the biopharmaceutical companies in really assisting them in their scientific endeavors to bring a drug forward. So some of the areas that I think are very exciting today about a career and research in the bioscience field relate to really, since the sequencing of the human genome has come about 20 years ago, is utilizing that information to get a much better understanding of the genetic underpinnings of many diseases. So it's been said, with the exception of infectious diseases, all diseases really begin by problems at the DNA level. So basically, when I say abnormalities, we're talking about mutations. So certainly, the role of understanding human genetics has been absolutely key and critical in developing many of the new drug therapies we have today and will continue developing in the future. It's also been key to another area that really is just on the cusp of being used significantly for treating human diseases, and that's, of course, gene therapy and gene editing. So, when you think about drug design in particular, one of the really hot areas of research these days is molecular modeling. So this really employs the kind of technology that will give us an understanding of the tertiary or three-dimensional structure of a target for a drug development program. So many targets for drugs are actually proteins or enzymes that play very, very important roles in the development of a particular disease. More recently, when we think about other targets for drugs, we're really beginning to think about the genes themselves, or the messenger RNA gene transcripts. So, but by and large, the vast majority of drugs that we have approved today really are targeting proteins. So one of the things that can help us immensely in designing drugs through in situ methods, including the technology associated with artificial intelligence and machine learning, rely on molecular modeling, understanding that tertiary structure, and then basically using computer technologies to actually design the program, virtually design a molecule that will fit into a binding pocket of a particular target protein to produce an inhibitory effect, for example. In some cases, it could be a stimulatory effect, depending on the disease you're talking about. So molecular modeling works very closely with drug targeting. So again, targeting, we're really talking about what are the processes that we understand about the pathogenesis of a disease. What is involved in this disease presenting, coming to the fore? And can we develop drugs that will knock out those processes, those proteins, those enzymes that play key roles in the development of a particular disease? So I already touched on gene therapy and gene editing. So, for the first time now, we have some studies ongoing globally where we're employing some gene editing technology as an effort to treat certain cancers. So again, if all diseases, except for infectious diseases, really begin at the genetic level, then if you're really going to cure these diseases in a permanent kind of way, then you're going to have to fix the problem at the genetic level. When we talk about targeting proteins that are involved in diseases, or enzymes, or when those enzymes or proteins are abnormal, what that means is we're talking about focusing on helping individuals with that disease by minimizing their symptoms. We're not talking about curing them. So that's quite a different thing. curing them, we really focus on gene therapy and gene editing. So gene therapies, kind of the progenitor for gene editing. It's been around for a number of years. It's really starting to play a more significant role in developing therapies to successfully treat a number of diseases. Basically, you're replacing a defective gene with a good copy or a functional copy of a disease that's involved in producing the disease. So this is going to be a very hot area, continuing to be a hot area, and I think this is going to be advancing very significantly in the next several decades. And probably someday, again, with the exception of infectious diseases, may actually have a significant role in replacing drug discovery and development. Immunotherapy, as particularly related to immune checkpoint inhibitors in treating cancer, is a very exciting area. I'll give you some examples of this in a bit. So the idea is that we have an immune system which functions normally to survey the cells in our body, identify those cells that have become cancers, and basically kill those cells, destroy those cells. That's part of the T cell immune response system that we have. Since when basically some cancers have means of defeating that process, and the immune checkpoint inhibitors actually are meant to prevent cancer cells from doing it. I'll give you an example of this shortly. Just want to touch a little bit then on this really exciting development about the messenger RNA vaccines that have been so successful in, bringing the COVID-19 pandemic kind of under control in a number of countries, and certainly as we get more individuals globally treated with vaccines, we should have much greater success in bringing the coronavirus pandemic under control. So I just wanted to touch on this technology a little bit. And I touched on artificial intelligence and machine learning systems a little bit already, but excuse me, one of the key things about one of the key things about all these technologies that are being developed has been the use of artificial intelligence, machine learning systems that basically aid in all steps of the drug discovery process. So this started about a decade ago. It's really beginning to take off pretty much in the biopharmaceutical arena. Certainly in academic research, basically to enhance our understanding of disease processes, and also to facilitate the decision making that pharmaceutical companies have to make based on the investigational drugs that they have, how to basically advance those down the drug development pipeline. So artificial intelligence, machine learning systems have assisted in those decisions and continue to take on greater roles, and certainly will continue to take on greater roles in the future.